protein is probably the single most important molecule of life. They're responsible for most of the functions required to sustain life and are manufactured within our cells, in our cells all the time. They're made of simple molecules called amino acids, which are chained together into long, intricately folded chains. And these are developed over millions of years of evolution. The structure of a protein is what determines its function. This makes comparison of proteins very important, and that's the topic of my research. Protein structural comparison is fundamental to humanity's understanding of things like biological evolution, drug design, degenerative diseases such as Alzheimer's and Parkinson's, and viruses such as HIV and Ebola, among many others. My research focuses on a specific type of comparison that biologists call alignment. As I mentioned before, this structures long chains of amino acids, and structural alignment is when, once overlaid, some amino acids occupy the same space as some in the other structure. You'll notice in the bubble at the top right, two strings of characters. Each letter represents an amino acid. Columns containing two letters represent two amino acids that occupy the same space. Columns containing a hyphen indicate an amino acid that doesn't have an equivalent in the other structure. To show this visually, notice in the box in the center 4 hemoglobin proteins on the bottom from 4 different species. From the left, a bloodworm, a horse, Kevin Rudd, and on the right isn't an alien, it's a sea lamprey. Notice how similar each of the hemoglobin molecules look. Most of their amino acids occupy similar spatial positions. Some of them not, but that will be reflected in the alignment. Unfortunately, despite 30 years of research in this area, current methods are quite suboptimal in comparing these alignments. They tend to subjectively weigh competing similarity criteria in a way that's not... They dramatically disagree with each other, often. I hope to remedy this situation by using objective measure of information content. What this really means is how long is the shortest message required to tell you where all of the amino acids are. Any advances in this area will result in furthering our understanding of all the things I said before. Thanks for your time.
Thank you.